Hello everyone. Our next topic in neoplasia is tumor suppressor genes. We are discussing about the cellular and molecular hallmarks of the cancer, and we have discussed about proto-oncogenes, and now the second is tumor suppressor gene. As the name suggests, tumor suppressor. They basically keep a check on uncontrolled continuously proliferating cells and prevent the development of tumor formation. We will discuss tumor suppressor gene in 7 categories. First is inhibitors of mitogenic signaling pathway. Second is inhibitors of cell cycle progression. Third is inhibitors of pro-growth programs of metabolism and angiogenesis. Fourth is inhibitors of invasion and metastasis. Fifth category is enablers of genomic stability. Sixth category is DNA repair factor. In the seventh category, we will discuss about some unknown mechanism that suppress the tumor formation. Now we will discuss about each and every category one by one. In first column we have gene, second the product of that gene, and in third column we have syndromes and cancer associated with the mutation of these gene. So first category is inhibitors of mitogenic signaling pathways. First gene is APC gene. Product of APC gene is APC protein. Mutation of this gene will result into familial colonic polyps and carcinoma of stomach, colon, pancreas, and melanoma, mostly GI cancer and melanoma. Second gene is NF1, neurofibromatosis 1 gene. Product of this protein of this gene is neurofibromin 1. Mutation of this gene will result into neurofibromatosis type 1, neuroblastoma, juvenile myeloid leukemia. NF2 gene, protein encoded by NF2 gene is Marlin. Mutation of this gene will result into neurofibromatosis type 2, schwannoma, and meningioma. Another gene is PTCH, coded for patched protein. Mutation of this gene will result into Gorlin syndrome, basal cell carcinoma, and medulloblastoma. P10 gene coded for protein of name phosphatase and tensin homolog. Mutation of this gene will be seen in Cowden syndrome and various cancer. SMAT2 and SMAT4, protein produced by these genes are SMAT2 and SMAT4. Mutation of these genes will result into juvenile polyposis and pancreatic carcinoma. So in the first category, inhibitors of mitogenic signaling pathway, we have discussed about the APC, NF1, NF2, PTCH, P10, SMAT2 and SMAT4 gene, their mutation and the cancer associated with these gene mutation. The second category in tumor suppressor gene is inhibitors of cell cycle progression. This category include RB gene and CDKN2A. RB gene encoded for retinoblastoma protein. Mutation of RB gene seen in familial retinoblastoma syndrome, retinoblastoma, osteosarcoma, carcinoma of breast, colon and lung. Other gene is CDKN2A, which encoded for P16 or INK4A, P14 or ARF. Mutation of this gene will result into familial melanoma, pancreatic cancer, breast cancer and esophageal carcinoma, melanoma and various leukemias. So in inhibitors of cell cycle progression, we have discussed about retinoblastoma and CDKN2A genes. Third category is inhibitors of pro-growth program of metabolism and angiogenesis. As we know, the metabolism is altered in a cancer cell and they require angiogenesis. So the more and more blood vessels supply nutrient for the cell growth. So these are the inhibitors of, in this category we will discuss about the inhibitors of pro-growth programs of metabolism and angiogenesis. So von Hippel-Lindau VHL gene encoded for VHL protein. Mutation of this gene will result into von Hippel-Lindau syndrome, renal cell carcinoma. STK11 gene encoded for liver kinase V1 or STK11. Mutation of this gene seen in Peutz-Jeghers syndrome or diverse carcinomas. All of these cancer we will discuss in systemic pathology in detail. Third gene of this category is SDHV and SDHD. This encodes for succinate dehydrogenase complex subunit V and D. Mutation of these gene will result into familial paraganglioma or paraganglioma. Fourth category is inhibitors of invasion and metastasis. CDH1 is inhibitor of invasion and metastasis. It prevents invasion and metastasis, so prevent cancer formation. CDH1 encodes for E-cadherin. Mutation of CDH1 will result into familial gastric cancer, gastric carcinoma and lobular breast carcinoma. Fifth category of tumor suppressor gene is enabler of genomic stability, gene that provide genome stability. One of the very famous example of this category is TP53, encodes for P53 protein. This is the most common gene mutation associated with human cancer. Mutation of this gene will be seen in Lee-Fraumeni syndrome, and this is associated with most human cancer. DNA repair factors, this include BRCA1, BRCA2, BRCA1 or BRCA2. Mutation of these will result into familial breast or ovarian carcinoma, carcinoma of male breast and chronic lymphocytic leukemia. Another group of DNA repair genes are MSH2, MLH1 and MSH6. They play important role in mismatch repair or microsatellite instability. We will discuss this in genetics in detail. So they encode for MSH2, MLH1 and MSH6 protein. Mutation of these gene will result into hereditary non-polyposis coli, non-polyposis colon cancer, HNPCC, colon cancer and some endometrial cancer. Last category is some unknown mechanism. Gene include WT1. Mutation of this gene is seen in Wilms tumor and some leukemias. MEN1 gene encode for menin protein. Mutation of MEN1 gene will result into MEN1 syndrome. Pituitary, parathyroid and pancreatic endocrine tumor. So these are the 7 categories of tumor suppressor genes that we have discussed. In other lecture we will discuss about the cell cycle and 2 very famous tumor suppressor gene like RB gene and P53 genes. Thank you.